Hello everyone, welcome to my tutorial. In this video, I am going to show you how to perform molecular docking using Autodock 4. Autodock 4 is a very important docking software and it is non-commercial or completely free. You can download this software from the website and you can install in different operating systems like Windows, Mac or Linux. So in this video, I am going to use aldehyde dehydrogenase 1A1 as protein and as ligand one approved drug telmisartan. Telmisartan is an anti-hypertensive drug used to treat high blood pressure. So using this protein and ligand molecule, I am going to show you how to perform molecular docking using Autodock 4. So these are the requirements if you are beginner in this field and if you are performing first time molecular docking using Autodock 4, then first you have to install Python in your Windows system and then MGL tool, that means molecular graphics laboratory tool. The full form is molecular graphics laboratory, and this Autodock 4 software is designed by the molecular graphics laboratory. So this MGL tool is actually a processing tool. This tool should be used to process the protein and ligand molecule. And finally, the Autodock application will be used to perform molecular docking. So these are the basic requirements for molecular docking using Autodock 4. So first we have to download this software from the website, and now I'm going to show you how to download and install this software or suite from the website. Okay. So first of all, type in Google Python, and this is the link for downloading the Python. Click here and you see the link for downloading Python has now been opened. So download Python 3.12.1. Okay, click here to download the Python. Next, type in Google MGL tool, and this is the link for downloading MGL tool. Click here. The link is opening, and click on the download tab. So you can see there are different versions for Windows, Linux and Mac. This is link for Linux, this is for Windows and this is for Mac. So this is the link for downloading the MGL tool. Click here to download the MGL tool. Similarly for Autodock, type in Google Autodock and click here download Autodock 4. Okay, link is opening and you can see several versions are there for Linux, Mac or Windows. We can click here to download the Autodock 4 for Windows. The softwares are now downloading. Then we have to install this software in our laptop or desktop. So you can see the three softwares have been downloaded. Click on Python and install it, but I am not installing because Python is already installed in my laptop. Similarly, for MGL tool, click here and click here yes. Click on next, then accept the terms and agreement, then click yes. So software will be installed. I am not also installing it because it's already installed in my laptop. Similarly, Autodock suite, you can click here, double click here to install the Autodock. Okay, so after installing, the default location of this software will be the C drive. Okay, so in C drive you can see there is program file x86, and here within the MGL tool, click on MGL tool, then lib and then site packages. Within site packages, Autodock tool is there, and click here and you can see within Autodock tool two dat files are there. Okay, these dat files are important. These are the application for Autodock tool, and copy this dat file and paste it in a separate folder. So I have created a folder with the name of docking. Okay, so I have pasted it, these two dat files within this docking folder. Similarly, for Autodock 4, we can see within the C drive there is a folder called Script Research Institute. Open it. Within Script Research Institute, Autodock folder is there, and within Autodock folder you can see two application, Autodock 4 and AutoGrid 4 are there. You also have to copy this application and paste it within this docking folder. I have created this docking folder on desktop. Okay, I have created separately because now on all the calculation will be performed within this folder. That's why I have pasted this application and this DAT file within this docking folder. And also the input files are present within this docking folder. And after performing the molecular docking experiment, the result files or output files will be saved within this folder. So this is the location which I will define within the docking software. So you can see here two input files are there, protein file and the ligand file. The protein is PDB ID of the protein is 7JWS. I have downloaded this PDB ID from the protein data bank. How to download the appropriate protein structure from protein data bank? To know that, you should watch my previous video on protein data bank. Okay. The ligand molecule, I have downloaded it from the PubChem database. You know, it's a tell me certain, as I have already told you. Okay, so I have initially downloaded it in SDF format, and then I converted it into the PDB format using OpenBubble. There is a separate video for downloading the chemical compound structure from different chemical database and on the different file format, how to convert them to OpenBubble. So you should first watch this video to gain some knowledge on how to download the protein and ligand. So now these two input files are present within this folder. Now I am going to process this protein molecule and ligand molecule through Autodock tools. So after installing the MGL tools and Autodock 4 software on desktop, you can watch this desktop icon. This is for Autodock tool. Now click on this icon to open the Autodock tool. Okay, so Autodock tool is opening. So this is the interface of Autodock tool. First of all, we have to define the location of the docking folder, because when I will select the protein molecules or ligand molecules, so this folder should be defined from where the protein or ligand molecule will be chosen, and also the output file will be saved. So click on preferences. Okay, so I have to define the location here. For that, click on the docking folder and copy this path. Copy, and then paste this path here. So make default and then set. So now Autodock tool identify this location where the input files are present and also the applications are present. Okay, now first of all open the protein molecule. For that, file read molecule. So you can see here the PDB files are showing only. Okay, so this first PDB file 7JWS is for protein. Open it. So the protein molecule is now open. You can see several water molecules are associated with this protein molecule. These red dots, these are the water molecules. Okay, and some ligand molecules are also present with this protein molecule. First of all, we need to remove all the water molecules and associated ligands and ions to clean this protein molecule. Cleaning is very necessary. Okay, so click on this 7JWS. You can see what are the structure or what are the atoms which are present under this protein molecule. So these are the amino acid residues. We start from aspartate 8, 9, then proline 10, valine. So scroll down to see the list of amino acids. So up to serine 501, this is the amino acid. Okay, then after serine 501, these are the ions, and this is the associated ligand, one chemical compound or known inhibitor. These are the ligand, ions, chloride ions, and then water molecules. These are the water molecules. Okay, so you can remove these water molecules from here. This option, edit, then delete, delete water. So all water molecules will be deleted. You can see now the protein is dehydrated, no water molecule is present. And if you click on this 7JWS, you can see now no water molecule is present. Only the amino acid and few ligands are there. So you can manually select these ions or ligands, because after serine 501, no amino acid is there. So these are the ions or associated ligands. You also have to clean this or remove this. Okay. So edit, click on edit, delete selected atom, because I have selected these ions. Okay, I have clicked on this checkbox and delete selected atom. You can see the only amino acid serine 501 is present. Up to serine 501, no ligands or ions are present. So now the whole protein chain is now clean. No water molecules, atoms, or ions are present. Okay, it's clean. So crystallographic structure also do not contain the coordinates of all hydrogen molecules. So first of all, we need to add all hydrogen molecules to make this protein explicit. Okay, so for that, click on hydrogen, edit, then hydrogen, add. You can add all hydrogen and also polar hydrogen, because mainly polar hydrogens are required for molecular docking, but you can also add all hydrogen. Okay, so after adding hydrogen, you can see the hydrogen atoms will appear. Okay, so these are the hydrogen atoms which were not present in crystallographic structure, but we have added it using this NGL tool. Then next, after removing the water molecules and after adding hydrogen, then we have to add charges. For protein, we need to add Coleman charges, and this is required only for Autodock or Autodock Xena. Different molecular docking softwares are there where these charges are not applied, or they have their own algorithm. But for Autodock, you need to add the Coleman charges. Next, next atoms, click on atoms and then assign AD4 type. Assign AD4 type atom. Okay. Now our protein has been processed. The protein processing is complete. Okay. Sometimes the proteins are dimeric or tetrameric or multimeric protein. So protein contain more than one chain. So for molecular docking, you always do not require all the subunits, because if the uh, active site or binding site is present only in one subunit, then you can select only one chain for molecular docking experiment. But if the binding site is present in between two subunits, where the amino acid residues from both subunits is involved in the formation of active site, then you have to retain the two chains. Okay, so first of all, you have to define the binding site. So depending on the location of binding site, you have to choose the protein chain. So in case of this protein contain only one chain, and the active site is present in the middle of this chain. So I have selected this protein. and the molecules can be docked within this binding pocket. Okay, so after processing of the protein molecule, we have to save this protein molecule in PDB-QT format, because when the protein molecule we use input in PDB format, but for molecular docking using Autodock 4 or VINA, the PDB-QT format is used. So save it in PDB-QT format, save, then write PDB-QT. So PDB file will be converted into PDB, and select all these, okay, up to end, add, and this, okay. Okay, so you can see the 7JWS is present in PDB-QT format. Okay, initially it was in PDB format. Now it has been converted into PDB-QT format. Now protein processing is complete. We are opening the ligand molecule. So click on ligand, input, open. So you can see PDB-QT file only is showing, but the protein is present in PDB file. That's why you have to select a PDB file. Now the ligand is also showing. Select this ligand and then open. Okay, so you can see the software has detected that this protein has 26 aromatic carbon and 8 rotatable bond. Okay, so you can see this ligand is present in green color. This is our ligand. Then click on ligand, torsion T, choose route. Then again ligand, torsion T, detect route. So the joining or root position has been detected. Now save this ligand in PDB-QT format. Ligand processing has been completed. Output save as PDB-QT. Okay, so ligand.pdbqt is PDB-QT save. So ligand is also saved in PDB-QT format, Autodock structure. Okay. Now, after processing of protein and ligand, we need to define the area where the ligand will search for its binding site. That means where the docking will be performed. Suppose if we define this area, only this area, a small area of the protein molecule, this surface, then the software will dock this ligand in this region only. So, okay, if we enclose this whole protein within a box, or we define this whole protein, then software will search for binding site on anywhere within the protein molecule. So we can define the area where the molecular docking experiment will be performed. Okay, so that's why the option is grid. We have to select the grid box. So first of all, choose macromolecule. We have to define the protein molecule. Here macromolecule is protein molecule. Select 7JWS, select molecule. Then okay, click on OK. We have to again change into PDB-QT. PDB-QT. Okay, save. It's saying should we replace it here? Click on yes. So now you can see the grid box can detect this PDB-QT format of this protein. Okay, now click on set map type. That means it's saying about the ligand molecule. So choose ligand. We have chosen the ligand. Then grid box. Okay, so you can see a grid box appears there. So if you start molecular docking, then ligand will be docked within this box only. But if you want to search anywhere within the protein molecule, then you have to enclose this whole protein within this box. So you need to increase the size of this box. So first of all, we are increasing the size of this box. We are enclosing this whole protein within this box. Okay, but it's better to select a small box size, because if the box size is large, so docking efficiency will reduce. Okay, and if the box size is very small, then there is a chance that few spaces will not be selected and appropriate docking experiment will not be performed. So it's very important to choose the appropriate box size. If you know the binding site or active site of the protein molecule, then you can define only that area. But if you don't know the binding site, then you have to enclose the whole protein within this grid box. So this box is very large. Okay. So these values 126, 126 and 126, these are the x, y and z dimension of the box. And these values x center, y center and z center defines the central position of this box. Okay, define the central position. That means you can shift this box. You can change the central position of this box. That box is shifting along x axis, then along y axis and then z axis. That means you can place this box. You can change the position of this box using this center x, center y and center z, and specifies the subspace. The dimension of the box along x, y and z axis is 70, and this is the center x, center y and center z coordinate. So now I am saving it. So close saving current. That means this parameter will be saved. Okay. Then we have to write a grid parameter file. That means output save GPF. This is grid parameter file. That means the coordinates of the center and the box size. This information will be saved in a GPF file. Okay. So name of this GPF file I am giving 7jws.gpf. I am saving it. So GPF parameter has been saved. Next, docking. So first we have to choose the macromolecule, then set rigid file name. Rigid file name means here the protein molecule, because protein molecule in molecular docking is usually rigid. They are not flexible, they are considered rigid. But the ligand is flexible. Okay. So here rigid file name is protein, and I am choosing 7JWS. This is the protein. Okay. Now we have to choose the ligand molecule. Ligand molecule, using ligand, select ligand. Okay. Then click accept. Now search parameter. So in case of Autodock is a Lamarckian genetic algorithm. So select genetic algorithm. Now you can see here number of GA runs. That means how many times the ligands will be docked into the search space. You can increase this number. You can increase to any number. That means 10, 100, 1000. So if you increase the number, more docking time will be required. More times the ligand will be docked. So automatically the docking time will also be increased. Okay. So I am keeping it 10, except. Then output. Output is DPF. That means docking parameter file. We have saved all the parameters. That means how many GA runs will be performed and everything. So it will be written in a docking parameter file, just like previously we have seen in case of grid parameter file. Here docking parameter file, and giving it the name 7jws.dpf. Okay, set. So grid parameter and docking parameter have been set. Now finally we are going to run the molecular docking experiment. First of all, we have to run autogrid. in Autodock there are two applications, Autogrid and Autodock. So first we have to run the Autogrid, then Autodock. Okay, run Autogrid. In case of Autogrid, first we have to choose the path name where the Autogrid 4 is present, and the path is already the path is the docking folder. So I am choosing here the Autogrid. Okay, this is the path of Autogrid in docking folder, and selecting the GPS, that means grid parameter file. It is saved in the docking file. Now a GLG file will be saved. This is the output of the Autogrid application. GLG means grid log file. Log file means the result file. So it is the result of the grid experiment launch. Now the Autogrid will calculate the parameters of the grid, and then a output file, which is grid log file, will be set. Again, this grid log file will be used as input for the final docking run. So it will take some time, and at the same time you can see in the docking folder some files are generating, these map files. If these map files are not generated, that means your docking run is not correct or some errors are appearing. So here you can see the map files are generating. That means everything is running correctly. Okay, grid run is now complete. Now we will run the Autodock. So again we have to define the path of this Autodock for application, and this application we have copied it to the docking folder. So I am choosing it from the docking folder. Here Autodock 4 application is present. I am selecting the Autodock 4 now. So application is Autodock 4. Then DPF file, docking parameter file, which is generated through Autogrid run. So I am selecting the DPF. Now the output file will be DLG file. That means docking log file, the result of the docking experiment. So DLG file is the output file or result file of Autodock 4 docking experiment. So I am launching it. That means docking run will start. Now molecular docking through Autodock 4 is running, and ultimately we get a DLG file. This DLG file will contain the scores of the chemical compound and also the binding poses, the PDB file of the binding poses of the chemical compound. The two kinds of output, as I have already told you in the previous video in basic concept of molecular docking. So if you are beginner in this field and if you are first time performing molecular docking experiment, better you first watch my previous video about basic concept of molecular docking. Then you should start performing the molecular docking experiment. Then everything will be clear. Okay, so at the final stage, at the end of the molecular docking experiment, we will get two kinds of results: the docking score of the chemical compound and the binding pose of the chemical compound. And we can visualize this binding pose in different visualization software like Biosphere Discovery Studio, Chimera, and Pymol. And I will make separate video on the visualization software and how to visualize the interaction. You can also visualize the interaction in MGL tool or Autodock tool, but it's better to use other visualization software. They are better in this aspect. Okay. Docking run is now complete, and it took approximately 8 minutes in my laptop. And if I had selected 100 GA runs, then it would take approximately 1 hour in my laptop, and it depends on the computational power of the laptop or desktop, and also the grid size. If the grid size is very large, then it would take more time. Okay, so now the result file is the DLG file. This is the output file or result file. So open it. It's a text file, and you can see, you can scroll down. Okay, this is the result file, and this is the first run number 1. Okay, and the estimated binding energy is minus 10.370 kilocalorie per mole. So first time when the ligand was docked, its score was minus 10.37. So total 10 runs were there. So let's see, and this is the pose of the first run. Okay, this is the pose. That means you can see the coordinates, xyz coordinates of the ligand atom. Next, this is the second run. Second time the ligand binding score was, or binding value was, minus 9.02. That means first time the pose was better. Third time the binding energy was minus 10.02. So total 10 pose and with their scores will be present in this DLG file. You can scroll down. Okay, and every detail you can see here: the intermolecular bond energy, dissolution energy, van der Waal, the contribution of different bond, and torsional free energy, everything, and ultimately neat binding energy will be present here. Next, run number 6 is binding energy was minus 9.89, and a table uh, will be present also. So the result will be tabulated to see the overall status. Okay, in this table clustering histogram, we can see, the run number 9 was the highest binding energy, minus 10.57, and this pose was repeated 3 times. That is, it is known as clustering. That means how many times the pose was repeated, or within similar pose were found. Okay, so 3 times this pose was obtained, this conformation was obtained, and with binding energy minus 10.57, and rest of the binding pose, these are the delta G value of the rest of the binding pose, and this pose was not very good because its binding energy is also low, minus 7.81. So the first pose, that means the pose with highest binding energy, was repeated 3 times. So if you increase the run number, suppose you have repeated 100 times the docking experiment, and more than 10 times, 20 times the pose was repeated with highest binding energy. So sometimes I get several questions: which pose should we choose? The pose which was repeated most of the time, or the pose with highest binding energy? In case of actually, here you have to choose the pose with highest binding energy, because it's about law of thermodynamics. Which pose is better is determined on the basis of the binding energy, only binding energy. The pose may be repeated 1 time, 2 times, 3 times. It can act as a checkpoint whether the pose was best pose was repeated or not. But it is not the main thing that the pose which was repeated most time will be the best pose. The pose with lowest binding, lowest delta G value is always the best pose. Okay, so this is the table format where you can get the detail of all the 10 docking experiments. And you see, the pose with highest binding energy was the docking was obtained in the docking run number 9. Okay, so we have to copy the coordinates of that pose, and we can paste it in a separate text file and save it as pdb. So this pdb file we can visualize in several docking visualization software. Okay, so number 9, 9 number pose. Let's see what is the number 9 pose. You see, this is the number 9 pose. pose which was obtained in the number 9 docking experiment, and its delta G value is minus 10.57. So we can copy this, copy the coordinate of this pose or conformation. Okay, so copy. So model 9, I'm copying it. I have selected because this is the pose with lowest delta G value. Now open a notepad and paste this coordinate, Ctrl V, by pressing Ctrl V, and save this data. Save as, I am saving it in desktop as based, giving a name based dot pdb. Always add this extension pdb, otherwise it will not be saved in pdb format. And select all files. Okay, then save. When you save, so you can see the pose is present here. So I am opening it in a visualization software, opening in Behavior Discovery Studio. You can also open the DLG file in the Autodock tool. Okay, so click on the docking, open the DLG file. Okay, ligand 2. Okay, so 10 dock conformation from 7JWS DLG. Analyze conformation play. Okay, so now conformation play. If I play this, you can play this number. This is number 1 pose, number 2, number 3, number 4, number 5. You can see. Okay, the green color is the original pose which was selected for docking, and you can see the poses are changing. First pose, second, third, fourth, fifth, sixth, seventh, eighth, ninth, 10th. So these are the 10 poses. Suppose the 9th pose is the base pose, so you can save it from here. This pose, write current. Okay, you can click here, write current, and save it as the pose 9.pdb. Save, close. So number 9 confirmation has been saved. You can check it here, the pose 9, and you can visualize in Behavior Discovery Studio. You can see this is the base pose or number 9 pose. Okay, so you can also open the protein here, desktop docking folder, docking here the 7JWS pdb, open. This is the protein molecule. Now click on view with hierarchy, and then pose view hierarchy. This is the hierarchy or detail of the pose 9. I am copying it, copying the coordinates. and pasting it into the hierarchy of the protein. Now you see this dock force is now present within the binding site of the protein molecule. This was the original ligand. I removing it and all the water molecule, and this is the dock force. You can see this is the pose 9. This is the dock force. It's now present within the binding site of aldehyde dehydrogenase 1A1. So you can also visualize the interaction, what kind of interactions were responsible for. So I will upload a separate video on the visualization and analysis of the interaction between the protein and ligand. Okay, so that's all for today. In this video, I have performed molecular docking using the Autodock 4, and we have saved the result in PDB format, the conformation in PDB format, and we got the docking score of the ligand molecule. Okay, so that's all for today's tutorial. Thank you.